cloning of whole animals. The completely whatever I discuss, it is completely like change. Like you can change the gear, so it will be in a different direction now. So two techniques are available: embryo splitting and nuclear transfer. As I told you, this time mostly I will use slide whatever present here and nicely animated so that you will not feel any problem. So if you see here, uh, this is embryo splitting, that cloning by embryo splitting. So after fertilization, the zygote is formed. I told you before, like when sperm and ovum they fuse together, that is called zygote. And that zygote can be divided into uh, like small, like from one cell to two cell, two to four, you will get like this, and finally it will from embryo. So as a result of that, that embryo is split. Like if this is the embryo, it can be split into two, and these two embryo can give rise to two different animals, but they should be the clone because they have everything same in terms of their genetic information. Why? Because as I told you, this is sperm and this is ovum. They fuse together, that is zygote. That means from one cell, zygote, it should give rise to one baby animal. But instead of that, after some division of this zygote, normally at around 8 to 16 cell stage, those embryo can be split into two. So basically that means those two embryo are coming from the same zygote, right? So as a result of that, they have exactly same genetics makeup, same genetic information. And now those two splitted embryo can be, implanted uh, in some surrogate mother, like in some other sheep, in this case sheep. So finally, from those two splitted embryo, you will get two sheep. So these two sheep are exactly similar to each other, so sheep A and sheep B. Why? Because they are the product of same zygote, and this is called embryo splitting. Now, this is very simple, right? And in the lab, not very simple in terms of understanding, it is simple, but in the lab, it can be done like this. The egg can be collected and fertilized in vitro fertilization approach, and then you can grow embryo in the lab, and then you have to separate those embryo into two, and then that embryo can be implanted in some surrogate mother, and as a result of that, clone animal can be generated. Here, I would like to mention something that is required to know. This is that embryo splitting, whatever I told, that can be done in lab, but naturally, sometime it happened. So, for example, you see some human twins, right? All of you know twin, right? So, what are those? I would like to mention here, there are two types of twin. One is identical twin and another one is non-identical twin. Identical twin are exactly same thing what I discussed, but that is happening naturally. That means after in human I am telling you. So when the zygote is formed, the zygote is dividing from one cell to two cell, 2 to 4, 4 to 8. But for some accidental reason, that embryo at early stage, they get divided into two. And both of these embryo, they are growing into two babies. But as I told you, they are coming from one zygote, so they are exactly same. They are identical twin. Now, answer my question. Have you seen identical twin and one of them, girl and one of them boy or not?
So
No,
that
sir.
can't be possible.
No, right? Yeah, no, it is not possible because they are product of a single zygote. right? So I understand that you can say no, boys and girls they look different. That's the that is phenotype outside thing, but they will be same because they are coming from same zygote. That's why their genetic constituent is exactly same. That's why identical twin always will be either two boys or two girls. But you have also seen non-identical twin. It can be one boy, one girl, two boys, but they are non-identical. What are those? Those are some again some some kind of aberration, not natural, natural but not very normal. What happened in normal cases? One ovum should be mature at a certain point of time. Ovum means egg cell, so that it can be fertilized by one sperm and then that will make one zygote and that will give rise to one baby. That is the natural or normal case. But in some rare cases, what happened at one point of time, instead of one mature egg, some female individual might have two eggs. They are matured at the same time. That is rare case, but that can happen. If two eggs are matured at the same point of time, so as a result of that, both of them, those two egg cells can be fertilized by two different sperm. So now two different zygote, and from those two different zygote, two different babies, they can be of same or different sex, but they have some genetic different, genetic constituent. So as a result of that, those are not identical. Someone now might be thinking, they say, why you are telling that genetic different? Because they are coming from same father, same mother. Yes, you are right to some extent, but yes, they will be different because I didn't discuss that yet. I think most of that, like in the next class, I will discuss. During the generation of sperm and ovum, some steps happen that is called meiosis. During that time, whatever genetic component we have, that get little bit, little bit like what I would say, some reorientation, some information coming from here and there, they are actually changing a little bit. Okay, during the process of making sperm and egg, during that time, that is called, that is called crossing over. So I will discuss in the next class. So as a result of that, it is two zygote. That means two different result of two different ovum, egg cell, or two different sperm. Then the zygote will be little bit different, although they have many similarity because they will be two siblings. So as a result of that, but they are not completely similar, right? So these are non-identical twins. So that's all. And if you have any interesting question, you can ask me. Otherwise, I'll go to the next slide.
Hi, sir.
Yeah.
Hello.
Yeah.
Sir, I was asking that can triplets be identical? Because when the embryo, when the ovum and the sperm mix, and when the cell division occurs, it's always in 2n number, like 2, 4, 8, then 16 cells. Then how? I mean, can triplets be identical?
So triplets, I would say, Anubhav. So the thing is, I think that if that zygote get distributed into 3 or 4 cell or even more, that will be lethal. That will not survive actually because the embryo should be splitted at very early stage. Then only it will survive. If the embryo is big and some problem happen, that will create problem. So as a result of that, like embryo should be splitted early stage only. I would say, Anubhav, in order to answer your question, as far as my current knowledge and understanding, this triplet can be done in lab, like whatever I told about the example of SIP, right? Although you have, say, for example, 16 cell stage, you can divide, like I would say, 4, not 4, like, 5, 6, okay, these cells. Because why we can split at that time? Because at this stage, all those cells, they have the, potential to make all different type parts of our body. I'm not going into that details. It will go into a completely different direction that is called stem cell and other things, developmental biology. But in the normal condition, I never heard that identical triplet. Okay. And
Okay, sir. Got it.
now coming into the next, the cloning of whole animal, this is nuclear transfer. The previous one was embryo splitting, that this is nuclear transfer. This is, as I I know that many of you are aware about the creation of Dolly the shape. Here, this is the procedure how Dolly was created. Okay, so I'm sure that many of you know that Dolly was a clone shape. So let's see how it can be created. I'm going step by step. Just go with me. Don't try to see everything. Then it will be a little bit problem for you. So this is a shape, cartoon diagram or schematic diagram. So from this shape, you can take just one cell. What kind of cell? That is, we are taking some somatic cell. Somatic means any body cell that contain the 2n information. That means the total number of chromosomes are present. That's why this is somatic cell. Say, for example, cells from some portion of this, this shape. So those cells are collected, and from those cells, you are taking just one nucleus. The nucleus is removed. So again, I'm telling you that nucleus have diploid information 2n. I'm not writing that 46 chromosome because this is shape. I don't know the chromosome number of shape. That's why I'm not writing, but it has the full set of genetic information. You have taken the nucleus from some body cell. At the same time, you are taking an egg from this egg cell. This egg cell contains the nucleus that is contain half information, the N. Therefore, when this egg fuse with the sperm, that sperm also contain the N information, half information. After fertilization, then it forms zygote. But this is a different experiment. So in this case, what we are doing? We are removing this nucleus. We are taking away this nucleus. This is the nucleus. We are taking away. So this is your, like in this case, this is the empty egg. Empty egg means what? Egg without nucleus. Now see what scientists did. They put this nucleus, which has been isolated from some body cell. It contains the full information. So as a result of that, they fuse this nucleus with this empty egg. And that's why this process is called nuclear transfer. Nuclear transfer. Now, this thing, right, the empty egg that contains this nucleus, it needs little bit of laboratory environment. It needs some little bit of electric current, and that will start the process of division. And then you will get the embryo. What you need little bit of electric application, I would like to say very briefly. Even in natural condition, when sperm and fuse, they fuse together, some potential difference happen in the membrane itself, right? Egg cell, sperm cell, right? So I'm not going into the details. In order to mimic that thing, you have to apply little bit of electric pulse, and then the division started, and then this embryo is formed. Then this embryo will be transferred into some surrogate mother, and then after like full pregnancy, then it will give birth to this little shape. Here, this is Dolly. So now, if I label here, I will ask you one question. If this shape is A, this shape is B, I'm just naming it, and this Dolly shape is D. Okay, can you quickly tell me whether this D will be exactly clone of A or B?
Sorry.
A.
Sorry, it has
Yeah.
nuclear information.
Sorry, A.
Yeah, very good. Yeah, you understood, right? Very good. Okay, so I'm successful. So you understood. I think that all of you who are attending the class properly.
Sir, I have a question here.
Question. The
Sir,
question is, yeah.
yes,
Yeah, Suma, you have
sir, the
question. Yeah.
tissues. Yes, sir. So the tissues will not be similar, like all the tissues in the body of the donor, tissue donor will not be same, like tissues are differentiated from each other. So will that not matter? Like,
Tanmay, yeah, good question. But if you remember that before lacoperon, I told you that in my body, in all tissue, we have same set of genetic information. The only thing in my eye cell, some set of genes are getting expressed. In my liver cell, other set of genes are getting expressed, right? So here what we are doing. Your question is very pertinent. But here what we are doing. We are taking the nucleus from some cells which will be easy to take. That's all. And then that nucleus, whether I am taking from my muscle cell or liver cell, but all this nucleus that contain the whole set of information. Now, whenever you are fusing this nucleus with the egg cell and you are giving that like some electrical pulse, you are basically reprogramming the whole thing. Reprogramming means it is again going into its initial stage. Now all genes are now active. From that again it will from embryo, because otherwise from our any other cell you cannot do that. That's why we are taking the nucleus and we are using that with the egg cell, the empty egg. That empty egg contains some constituent. I'm not going into that details. Some things are present in empty egg. That is all that is required, absolutely required. Otherwise, nothing will happen, right? Even for natural fertilization, what happened? The egg is there. Egg contains a lot of other things apart from the nucleus, and from the sperm only the nucleus is coming, right? So similarly, we are mimicking this thing. We are taking the nucleus from body cell. We are fusing with the empty egg, and we are then after this short electric pulse, it will be reprogrammed. Then that means reprogram means all those genes will be again active and it will start working, and slowly it will be differentiated, and then from embryo it will get up like full grown baby. Tanmay, is it clear?
yes, sir. So
Yeah.
all the like all the nucleus in all our cells have the same genetic setup, but the expression of genes are regulated in different tissues. That is why we
Exactly,
get
exactly,
different.
exactly. That is happening during development when we are inside our mother womb, like when we are getting developed. I would say after 10 weeks. This week might be wrong because I'm not a teacher of developmental biology. But the thing is, slowly, heart is getting developed before our birth. So then kidney, all those things. That means those kind of like specialization happening. That some of those organ formation, all those things are happening before the birth. So they are getting specialized. But at embryo stage, all cells they have the potential. Their genes are expressed. They can express everything. But slowly over the time, they are getting specialized. Okay. But only thing here we are taking nucleus from already differentiated, as you have correctly mentioned, some cell, but we are reprogramming it so that it can be again it can give you embryo, and from that embryo this baby can be generated. Okay, but I would say very good point actually. So this is Dolly, and this is the schematic diagram. But here you can see the actual one. This is Dolly, born in 5th July 1996 at Roslin Institute, Edinburgh in Scotland. And this is the process. Whatever I told, I discussed in the last slide, exactly same. Only thing here mentioned in case of Dolly, cells are taken for nucleus from the memory cell. Memory cell, right? Because of I think nothing. They just took some soft tissue so that they can take out those, nucleus, in a better way. But that I believe people can take from other cells also, right? Memory cells, right? Diploid cells like normal body cells, and from there they took the nucleus, and from the egg cell they enucleated. That means the nucleus is taken away, and then they fuse it, then they add some or they that provide some current pulse. The physics and chemistry behind this current pulse, I am not very much aware about this, but as I told that even for normal fertilization this potential difference happened during this fusion. And then this dolly was created. I would like to say the dolly was healthy enough actually, and it was dolly was surviving many years, and dolly also reproductively healthy. So Dolly has, next generation also. And at the end, it developed some autoimmune disease, arthritis, and other things. It was in some zoo, and finally Dolly passed away. And anyway, but it was a successful experiment. Okay, so I will only few slides I have, but those are very interesting slides. So I'm just going through those things, but I will not
Excuse me,
discuss.
sir.
Yeah, sure.
Sir, if all the content in our cells is the same, so does that mean that no new cells is added after some initial few months after the zygote happens, takes place?
Yeah, no new cells means, yeah. So the
Means
genetic
only
information,
through mitosis have new cells are formed, not
only
through.
mitosis, like new cells are from exactly same genetic component, only in germ cell. That means when the sperms are produced in case of male and ovum or egg cell in female, during that time some genetic rearrangement happen only in sperm and ovum. I didn't discuss that yet, but you might have studied in your class 9, 10 or 11, 12, if you can remember a little bit, but I will discuss to some extent during my next class. But those are only germ cells during the sperm formation and the egg cell formation. Otherwise, all cells, the divisions are mitosis, and they are just making like from one cell to two cells. But before our birth, during the developmental stage, those cells are getting specialized. Some cells, they are specialized to make digestive enzyme. Some are like heart muscle, all those specialization. But if you sequence their genetic component, their DNA sequence will be exactly same because they are coming from the same zygote. Clear, Rajesh?
Yes, sir.
Okay. Okay, good.
Excuse me, sir.
So next slide, please. Anubhav, you are asking something?
Yes, sir.
Yeah,
Sir,
so the sound is
this
very
may be
low
a
actually. Yeah, little bit loud, please.
sorry. So this may be a very silly question. Like during cloning, we implant the nucleus into the egg cell and then we place it into a surrogate mother. Sir, so what if instead of using a surrogate sheep, we use some other animal of the sheep family and place the newly formed zygote inside that animal? So will that also give rise to a successful cloning experiment?
Yeah, so yes, so Anubhav, your question is not silly, it's a good question, but maybe beyond our discussion, but it's a very good question. So for example, even in sheep, they use different types of sheep. In case of Dolly, they have some Scottish sheep, some black-faced sheep, it is possible. But Anubhav, the problem is if the species and everything is completely different, then it will be some compatibility issue when the baby is growing up in a mother's, uterus, like the problem will be some compatibility issue. But as you have seen in this case itself, like if you see, this is Scottish blackface. From there, they have taken this, like empty egg, and Finn Dorset, they are looking different, right? From their nucleus is taken, right? And then they put it in another like type of shape. So yes, it is possible, but you have to be careful about that selection. Definitely, if it is within the species, within the same group, definitely it will be much easier. But anyway, this experiment itself is not easy. But yeah, your question is good, not a silly question, I would say.
Thank you,
And
sir.
now the next thing is the first clone cat. So in 2001, scientists at Texas Austin University, so they created the first clone cat. Here, these are some stories just for you actually, So here, she is the owner of this cat. So this is clone cat. Also, you can say copycat because I think you get the sense why copycat, because this is clone, exactly same thing, right? So that's why. So, but anyway, this information you can right now you can forget, no problem. But I want to see not only shape, other mammals are also being cloned. This movie you might not see. This is like before your birth, like in 1997, it has been released. I have seen this movie. Okay, so this movie actually tells about the selecting best possible features from parents, the science fiction film, and how people use this, the modern, like powerful technology of genetic engineering in order to select like best features from father as well as best features from mother. Again, I'm telling you, this is nothing but science fiction movie, not the real thing. But anyway, but this movie was, released in 1997. But I think you have seen, like I just, I'm not showing you the slide, but have you seen the movie name there? What is written there? I'll show you, but intentionally I'm not showing you.
So a DNA
Yeah,
nucleotide
very good.
sequence.
Nothing but this is nucleotide sequence. The movie name is Gattaca. This is not Gattaca. This is actually GATTACA, nucleotide sequence. Right? The movie name was like that. And you can understand this is too much science fiction movie. Maybe this was not a very hit movie, but it has some useful information now at this point after attending science of living system class. If you see this, you will understand most of those things. And here also, if you see this spiral staircase, this is nothing but DNA. This is some in the background you can see some DNA like structure, and this is some nucleotide sequence like Gattaca. So anyway, this is just to tell you the first clone primate. As far as whatever I told you, that is like cat and sheep, those are mammals, but not in the primate. That means not very close to human. But this is close to human, some monkey, right? Their name Zhongzhong and Huahua. They born in, I believe, in China actually in 2017, right? And this is crab-eating monkey, and this is again the first clone primate. Okay, and now the next slide is problematic slide, but again that is in China, and the scientist is now behind the bar under prison. So the thing is, what he did, he actually, I would say, unethically edited some of the genetic makeup of human babies. There are, whatever I discussed, like genetic engineering, cloning of gene, those are fine. You are making some product, useful product in bacteria, in some other cell, yeast cell, for mankind, right, for our own benefit. But even in order to do that, we need to take some ethical clearance. That is true for even for in our lab also. When we do this thing, when we are applying to funding agency for the fund for money, we need to provide this ethical clearance that what kind of experiment I am going to do, what are the risk and benefit, all those things. So as a result of that, you cannot do everything everywhere. You need to strictly follow some rules and regulation. Those are actually, imposed there. Now the problem is this scientist in China, he created, his name is he also. His name is he, something like, I forgot his name. So the full name I'm saying, that he created like those two baby, Lulu and Nana, those two are human baby, but somehow he modified its genetic constituent. Now the problem is after that, there are a lot of criticism in newspaper. You can see this is in 2018. Experiments to gene edit babies are criminally reckless, says Stanford bioethics community. And here in BBC News, gene editing babies called to pause humanity altering research, because it will create a lot of problem also. That part you have to understand, right? So as a result of that, there are a lot of deliberation, and finally that person is, as I told, behind the bar. And anyways, so these things are good when you are actually doing for good, and if you are using in a different way, then it is bad. So this one, the last slide. So both of these scientists, they got Nobel Prize in 2020, right? Just last year. So when I used to teach in 2015 and 16 in the class, I also used to teach this thing, and I used to tell students that they are going to get Nobel Prize within a couple of years. Last year they got it. I'm not going into the details of their discovery, but what I want to say, they actually, I would say, they make some kind of devices, or they discover something so that you can edit genetic information more precisely and more quickly. Instead of restriction enzyme, they use some Cas9 CRISPR-Cas system. So some of you might be aware that CRISPR-Cas system. Again, I'm going to tell you. I'm not going to discuss in details, but this is some very modern technique to edit genetic information, and it is very user friendly. We can use in our lab also. But the thing is, because of their discovery, they got Nobel Prize, both of them in 1920. And that's all about genetic engineering. And you know, uh, and I believe that I don't have slide. Just thank you. So anyway, this is done. If you have any quick question, you can ask me. And also you have class, right?